Okay, so a wee bit of greater detail when it comes to DNA replication. So we looked at the last video we looked at the kind of simplified diagram of how this molecule, this double helix, likes to unwind. And then the two strands are going to open, okay? And three DNA nucleotides are going to come along and create a new strand, all right? But we have to introduce the components and the importance, the requirements of different molecules to actually make this happen, okay? So again, using the exact same colours and everything for ease of understanding, here is our DNA molecule, okay? And as you'll see, it is running anti-parallel, 3 end to 5 end, and then the opposite strand, okay? So if that's, so this is the template strand here, the original strand, okay? 3 to 5, and then the opposite strand for this molecule is going 5 to 3. So these here, these two black lines are your template strands. Okay, that's what we're going to copy. We're going to synthesize a new strand from as such, okay? And then before we start, I want to kind of introduce you to the kind of the importance of what we need. So for us to basically unwind, okay, and open these strands up, okay, we require DNA polymerase, okay? So there's two types of enzymes involved here. There is DNA polymerase and there's also ligase, okay? But essentially all you need to know is the DNA polymerase basically is responsible, so according to the SQA, responsible for opening up this strand, okay? So we're going to class this bit here as the replication fork, okay? We'll talk about that in a greater detail in a wee minute. So this is the direction and how we're going to replicate new strands, okay? So first thing is, requirements for replication is we need a DNA template, so we need a DNA molecule, yep, got that. DNA nucleotides, okay, so these 3 nucleotides, so adenine, thymine, guanine and cytosine, okay, do we have them within the nucleus? Yes, we do, okay. And then we need a primer. So a primer is what's classed as a pre-existing chain of nucleotides, okay? And the way I try to kind of explain it, obviously the girls get it more than the boys, but a primer, if you imagine it like a primer for makeup. So what happens now is there is makeup that you can put on, so you clean your skin, you put your moisturiser on, you know, to make sure there's hydration in your skin. And then what will happen is some females will put a primer on. So again, it's just a cream or a gel that basically helps set the foundation. So when they start actually putting the makeup on their skin as such, so they need it to start the process of applying their makeup. So it's the exact same for DNA replication, okay? We need a primer, okay? So it's a short chain of nucleotides to kickstart the process, because our enzyme DNA polymerase can only add nucleotides on, so create a new strand, if there is a primer present in the first place, okay? And then the final thing is we need ATP. So anything we are making within the body, if we're synthesizing something, we need energy behind it. And as we've already talked about a couple of weeks ago, we require ATP, we require energy for this process. So there'll be lots of ATP molecules circulating around. Okay, so let's kickstart. So this molecule has been opened up by DNA polymerase, okay? And what's going to happen is we're going to take a copy of this strand and we're going to take a copy of this strand. But we're going to do it in two ways, because there is golden rules to replication. Okay, so when we talk about DNA polymerase, okay, DNA polymerase can only add new nucleotides to the 3 end of a chain, okay? So with a new strand being synthesized, the rule to remember is that DNA polymerase can only bind on new nucleotides to the 3' end, okay? So this is why this primer here, so remember this pre-existing chain of nucleotides, okay? So what's going to happen is that's going to come along and it's going to bind on to the 3' end of the template strand, okay? Remembering at all times that if this is the template strand, okay, this is the 3' end, our strand, so again our final rule, is always going to have to be replicated in the direction of 5 to 3, okay? In this strand, so our leading strand, so if the way to remember it is a leader is a person that goes forward and is meant to inspire somebody, etc, etc. So the leading strand is always replicated first, okay? So this here, so this class is this replication fork, because essentially remember this molecule is a long, very, very long molecule. So what's going to happen is there's just going to be sections, so it's almost, if you imagine, it's called replication bubbles. So if you see this as our strand, so here's one strand of our molecule, there's the second strand in our molecule, and what's going to happen, this replication bubble, is this DNA polymerase is going to go along and open up this molecule at certain sections and replicate that, and then it will close, and then it will keep on moving. So it moves all the way up, because eventually, if we could, you know, do this in real time, what would happen is this would end up just replicating and opening constantly all the way along. All these two strands are going to be copied, and then it basically, these two template strands will close down again as the new strand is being made, okay? So it's a long, long process, but when it is being replicated, it has to be replicated in the 5 to 3 direction, okay? So our leading strand here is basically, so the leading strand goes towards the replication fork, replicating in one direction only. So this is basically the direction that the DNA polymerase likes to travel, okay? So we have our primer to kickstart the process, and then what's happened is DNA polymerase, it's basically started here, okay? And what's happened is it goes along, creating the new strand, adding on the new nucleotides, okay? And the new strand is replicated from 5 to 3, okay? So DNA polymerase starts, so the primer starts 3' end, DNA polymerase is coming along and adding on the nucleotides from our 3' end as well, creating, as it's going along doing its job, till it reaches the very end, a new strand that's synthesised from 3 to 5 direction. Again, remembering this is just what we're talking about, the anti-parallel, okay? But there's a difference. So the leading strand's nice and easy, okay? But there's a difference with the lagging strand. The lagging strand's a wee bit more complex, okay? Because remember, it doesn't matter whether it's a leading strand or a lagging strand, we have to go by with the replication rules. And this is anti-parallel. So this is not nice and easy where we're just going from 3 to 5 from the template strand, okay? Because the opposite strand is going to be 5 to 3, okay? So what happens is this lagging strand, okay, and if you again try to simplify it, if something's lagging, it's lagging behind, it's going slightly slower. So this is what happens with this strand, okay? So essentially what's going to happen is a primer is going to be laid down, okay? So a pre-existing chain of nucleotides is required, and that's going to be laid down at our 3' end, okay? And then, so this is a 3' end here, sorry, right? We say, so primer here, okay? So the primer's laid down, and then what's going to happen is our DNA polymerase comes along and starts to synthesize our new strand. But essentially what's happening is, as it's synthesizing, okay, it's basically running out of template, okay, because again, it has to, the DNA polymerase will only add new nucleotides onto the 3' end, okay? So what's going to happen is it's going to eventually run out of template, okay? So it eventually goes back and starts again, so it creates another wee primer, So what's going to happen is we're going to have individual fragments being synthesized. Okay, so it runs out of template and then it's going to go back along. Okay, it's going to lay down another new primer, and DNA polymerase is going to come along and add on our nucleotides again, and then it's going to run out and it's going to have another primer attached. And you're literally talking about they're doing it only in chunks of 2 or 3 nucleotides at a time. Okay, and then as I say, what happens is, and again I've simplified this, so I'm just showing this is large, this is only 4 nucleotides here, to create to show you each fragment. Okay, but the lagging strand, as I say, has these fragments. Okay, and what's going to happen is you see this in a really nice animation. So on YouTube we're uploading these videos. There's also videos that obviously other people have made with animation in it, and you'll see a nice video on what happens is eventually, so as these fragments are being synthesized, the primer comes away and new nucleotides starts binding on to this pre-existing fragment already. And then eventually what happens is we can't have any separation within this strand. It has to be one continuous strand. So we get this addition of this other enzyme, ligase. And it's almost like a glue. So a classic SQA question is what molecule is responsible for joining the fragments on the lagging strand, and it's ligase. Okay, so eventually ligase is going to come along and join these fragments together, so it creates one like this, one nice new strand. Okay, but again, going back and remembering the golden rule, so as we can see that a new synthesized strand is replicated in one direction only. Okay, it goes from 5' to 3', and it's the exact same. You can see this by these arrows. It's exact same for the lagging strand. Okay, it's going from 5 to 3. So primer, nucleotide, so DNA polymerase will come along, add on, stops another fragment, runs out of template. So here's another little primer, comes along, DNA polymerase binds on the nucleotides, and then we've got a series of fragments. Ligase comes along, adds on the fragments, and then we've got one strand. Okay, other words you'll probably find if you're reading up further about this is the leading strand is classed as continuous, the continuous strand, i.e. it's continuously synthesized and built, whereas a lagging strand is classed as discontinuous because it's not continuously built, it's built in fragments. But the most important thing is the fragments are joined at the end with ligase. Okay, so the requirements for replication. Okay, DNA template, we need our nucleotides, our free nucleotides, we need our primer, we need our pre-existing chain. DNA polymerase needs that there to start this synthesis of our new strand. We need 2 types of enzymes, DNA polymerase and ligase, and we obviously need ATP, we need energy. Okay, and then the most important thing, and this will get you through, because some people will say that you know, Miss, this is really, I don't understand this, I find it quite challenging, etc, etc. See if you just remember this, because essentially no SQA question will ask you to replicate a diagram like this. Some pupils, you know, you can, if you do an extended response question and it's in relation to the replication of a DNA molecule, you can absolutely, if it, you know, if your learning styles that you prefer, I'm very much a kind of visual learner, so I prefer diagrams, and as you can see, colour and etc, etc. But if you could put this diagram down, as long as you annotate it and you explain, it's not enough just to throw something down like this. You would need to be putting your little notes at the side to explain the process, okay? But if you're still struggling with that, if you remember these golden rules, these replication rules, that'll be enough to get you through any form of question, okay? And the 3 things are: DNA polymerase adds nucleotides to the 3' of the new strand, really important, okay? Primers bind to the 3' of the template strand. We need a primer. DNA polymerase needs a primer to start the synthesis of the new strand. And then finally, replication of new strands is always in the 5 to 3 direction. And when we talk about 5 to 3 direction, we're talking about the synthesis of the new strand.